Okay, so I'm going to pick it up from where Town left it with the BAM diagram. And I will say before, this is a lot to ask, but before you jump into running your models in R or running your Maxent, think about the BAM diagram. And I just, instead of having, I just artificially placed a map. So think about the geography, think about where your organisms may be and where these three important dimensions, abiotic, biotic, and movement, where these dimensions affect the species in geography. So think about that. Like I said, it's a hard thing to ask, but make an effort to think about that before you run models, and think about the biology of the species to the best of your knowledge and the best information available. I know of a student, I teach a niche modeling, species distribution modeling course, a grad course for 1 semester. I had in, I think this is maybe, I've taught it 5 semesters, not all here, just 1 semester here, but at Oklahoma State, I had 1 student in those 4 or 5 semesters, who thought about this and designed his study based on the BAM diagram. And I was very impressed because it's really hard, it's hard to think about this conceptually before we run models. But the reason we should do that is, well, first of all, because our fitting of the model will probably be will be better, but also it makes us think about what are we getting out of the model. So we are getting a geographic region, an estimation of potential distribution in geography that can be the occupied distribution, so the intersection of those 3 circles, or it can be as broad as the abiotic conditions that are suitable for the species, so that the whole A circle in geography. So this depends on the configuration of a BAM diagram, and Town mentioned different species will have different configuration, generally species versus endemic species versus species that have no dispersal abilities or low dispersal abilities versus, I don't know, continental dispersal abilities. And then how well we sample the environment space, is it uniform or not, and what algorithm we are using. So think about these concepts and these constraints of your experiment before you run models. Okay, so what do we do? What is the practice of niche modeling? So we have a study area, so practically thinking about modeling, ecological niche modeling. We have a study area, which is a raster map, and we should think about what is the extent of our study area, because that affects the model outcome. So if I have a species that is distributed, or the known range is southeastern United States, I should probably not use as study area whole North America, because I am sampling environments that this species has not reached, because it does not have that dispersal ability, or because biotic interactions prevent the species from dispersing. So think about study area before starting running the models. Another difficult question, another difficult aspect of modeling. What should be the extent of my study area? Then the dependent variables. Dependent variable is obviously species presence data. But think again back at the BAM diagram, where are these presence data coming from? Are these presence data coming from the occupied, the intersection, the perfect storm of those three, B, A, and M? Or are the presence data coming from sink populations, meaning populations that are not viable long-term because of maybe lack of maybe because of a competitor or a disease, so on and so forth. So we have occurrence data, but we should ideally think about where they are coming from in terms of geography and relationship to this BAM diagram. Then we have environmental variables that characterize each cell of that study area, and then we need a function. That's basically what we are doing with modeling. We need a function to connect our dependent variable, the species presence or presence absence, to that environment, to those environmental variables we have that characterize our study area. So the response is presence of species. The explanatory variables are, and this goes back to abiotic versus biotic. So the explanatory variables for presence of species for this response are variables that we observe and estimate, for example, temperature, and variables that are not that easy to estimate, and by that we mean biotic interactions. So ideally, we should have a function that incorporates both easy to estimate or easier to estimate and not as easy to estimate the biotic interaction. So think about having a surface, a raster, a map of biotic interactions. That's quite difficult to obtain, right? So because that is problematic, because of the biotic interactions are problematic to quantify over long periods of time and large extents at fine spatial resolution, what we do, we approximate that response, that presence of species, we approximate, or presence absence, we approximate as a function of the variables, environmental variables that we can observe and estimate or quantify. Okay, so the general approach is we calibrate the model. So we estimate that function using available data or species data and a set of variables. Then the model provides us with a prediction over a set of variables, values over the study area, right? So the calibration is done at the locations for which we have species presence or presence and absence. We use the environmental variable, the environmental values at those presence or presence absence location. Then we make a prediction over our entire study area, so let's say Southeastern United States. But then we also need to evaluate the model. And the evaluation should be done and is done with observations that were not included in calibrating the model. Otherwise, it's a verification and not a validation, which is what I'm saying here. It's a verification and not a validation of the model. So we want to validate the model with data that were not used to calibrate the model. Otherwise, we are just verifying the model, not really testing the model, which we do with the validation approach. Okay, so any questions? Yes. Do you mind using a microphone?
Hello. Yeah, I'm just wondering how many species we talk about. Like, is it like two species or like taxa or like
what
this modeling?
So the models are run species by species, but you can run hundreds, thousands of species.
Oh.
There is no interaction of species in the modeling process, in the algorithm. So you do individual species modeling, but multiple, as many as possible, as you need. Anything else? Okay, oh, yes. Microphone, I'm sorry.
So basically, the calibration area is M, no? So you need to have that M defined so that you can use that background within M to calibrate the thing. So that's why it's really important.
Yes, exactly. And if you calibrate, we'll talk about, I think at the end of this presentation, I have how you evaluate the metrics to evaluate models, and some of you, I'm sure, are familiar with these. When you use a very large area, you are artificially inflating your evaluation metrics. So we'll talk about that. That's why it's important to think about that M. And I'll explain that in a minute. Anything else? Okay, so very quickly, going through types of algorithms. So besides the maxent that we will use, there is presence only, which basically represent envelopes, environmental envelopes around data, presence data that we have access to. So these are methods like BioClim, Mahalanobis distance that have used the multi-dimensional, not just two, but I'm showing two environmental dimensions, and then presence points, and then fitting an envelope that can be as simple as a square or more tightly fitting presence data. So this is dealing with, I have only presence data, I don't have anything else. How can I come up with some estimation of what is suitable for the species and what is not suitable for the species in this environmental space? Then we get to presence absence. So now we have both presence data and absence data, and we can do regressions. And there are many options: general linear models, general additive models, classification and regression trees, so on and so forth. But this approach, this modeling approach, should be best. Its use would be best when we try to estimate, when our goal is to estimate that occupied distribution, geographic distribution. So the intersection of the three circles where the species are present. If we have absence data, this would be the best, I would say, and not just me, but some of us, some of us in the field would say this would be the best approach. Now, talking about absence data, I think Town will talk about maybe absence data is another whole nother can of worms. What is absence, detection, biotic interactions, all that. But if we are in a good situation of having both presence and absence, regressions are the best, and it allows us to get very, in theory at least, very close to that observed occupied distribution of the species. Then we have presence background, and this has been the star of niche modeling in recent years because of Maxent. I used to have a graph showing how often the first Maxent paper was cited. It was published in 2006. It has been hundreds and thousands of times cited. So this method has become very widespread in use. And that is because, we only need presence, we don't need absence. And a lot of times, at least in my case, a lot of times I find myself, most of the times, I find myself in the situation of having presence data but no absence data. So in this approach, what we are doing is we are taking known presence data, and then we are sampling the entire extent of our study area. We are sampling the environments, and we are trying to, and again, this is in one dimension, but we have multi-dimensional environmental space. But what we are trying to do with this presence background approach is figure out which conditions, which environmental conditions are more frequently associated with presence than the background. So we are trying to separate between environmental conditions, combinations of environmental conditions that are associated with presence versus the rest of our study area. We're not saying it's absence. A lot of it is absence, but not all of it. So this is the approach. If you don't have absence data, this is the approach that has been used for quite a while, frequently in the last 10 years or so. But there's also ENFA that precedes Maxent, but for some reason, Maxent is pretty much dominating the field right now in terms of presence background. Then there is presence pseudo-absence, which is pretty much presence background, with the exception that with presence pseudo-absence, the pseudo-absence do not include pixels that we know have presences, have known occurrences of species. So background includes the entire, randomly samples the entire extent of our study area, whereas presence pseudo-absence randomly takes pixels that are not associated with any presence of the species. Now this is a minute difference, and usually in the field, in the publications, you don't see this difference. It's minute because think about 1 kilometer resolution, again, southeastern United States, there are millions of pixels, and I have a species with a known presence data set of 100 if I'm lucky. How likely am I in the with presence background versus presence pseudo-absence, how likely am I to hit those 20 or 100 presences out of 2 million pixels? So the difference is small. It becomes significant in certain situations. So in general, we do not separate between presence background and presence pseudo absence, but you will find this in the literature, this separation. Okay. For some reason, the screen and the projector are starting to, they also need a break. Not my screen, just this and the projection. But so focusing on presence background models, very quickly, what do we want our models to give us? A correct prediction of known presences. So basically, if I have presences, I want that model to be able to predict those presences as present and not absent. And then we also, that's why we are doing this, we also want some sort of suitability prediction for the rest of the study area. So if I have a model that has maybe 100 occurrences scattered across Europe, I run a model, I want those presences to be predicted present, but I'm also after this potential distribution or predicted suitability across the extent of my study area. So I have high suitability versus low suitability, varying degrees of or shades of gray. Okay, so when are models failing? Models are failing when we have presences that were incorrectly predicted as absent, and that means we have omission error. I'm going to try to refresh this screen because it's bothering me. Oh yeah, the whole screen is going a little crazy. So we have expectations or models failing, not failing, but not performing well when known presences are predicted absent, and we call that omission error. And when we have a very broad distribution, potential distribution across our study area. So that means commission error. That means a lot of the unknown space is predicted present. So here's an example of commission error low. This model has a limited or a more contained potential distribution. And then commission error high, pretty much the entire Europe is predicted present for this species. So we'll talk more about this, but we know we have this problem in presence background. We don't have absences, right? So we cannot say this is bad or that is bad. We don't have a way of really quantifying that because we are dealing with background, not true absences. So we have to take into account the fact that we have commission error, but we don't have a specific way of measuring this commission error in the presence background models. I'm going to stop here because it's already time for coffee, and because the screen is going crazy, and because we are reaching confusion matrix, which is extremely confusing to everyone. So without a break, I think everyone will break down looking at this confusion matrix. So. Okay, so confusion matrix. I think people are still filing in. The one funny story, at least in my mind, the one funny story about confusion matrix is that I gave a pop quiz in one of my classes, maybe the second time I taught the grad course, and I asked the students to draw a confusion matrix and explain the 4 boxes. And I think it was a D overall, D average in my class. So it was pretty bad. And then one of the students who took that class, I was on his committee when he had his comprehensive exams. I made it an open book exam, and I made him do this. So he had fun with I think he had, maybe not. Maybe he was pretty annoyed with me. But what is confusion matrix? We are just summarizing actual presence and absence versus predicted presence and absence. And that's how you start the confusion matrix, actual versus predicted, and you have presence, absence, presence, absence. And then within this, you have 4 combinations, right? So you have present, actual, and predicted present, present, actual, predicted absent, same for the other actual versus predicted absent. So based on that, we calculate omission error rate, which is how many actual presences were predicted absent versus the data set of presence. So that's omission error rate. Also called false negative rate because we falsely assign absence or negative to presence data. And then commission error rate has to do with absences, and it is absences that are predicted present over known absences. So you don't have to recite this in your sleep or when you are startled, but know how to draw it and then know how to make sense of it. And you'll see why that it comes, well, first of all, omission error rate is one metric of a model evaluation. So we need to make sense of it. And then commission error rate comes in later. There are also more calculations. Also, we come in 2 slides will be important. Sensitivity versus specificity. So sensitivity is how sensitive, how good is our model at predicting presences. So it's presences over known presences. Predicted present over known, all known presences, and then how specific is our model, meaning predicted absent over all known absences. So this is model is failing, this is model is doing good. So we have error rates, omission and commission, and then we have model sensitivity associated with presence data and model specificity associated with absence data. Again, all you need to do is draw that matrix, and that's all I wanted my students to be able to do. And I guess I failed as an instructor because they were not able to do this and then recover the problems versus good outcomes of the model. Any questions here? Yes? Oh, microphone, I'm sorry. This is being recorded. Whoever had the last question has to walk the mic.
I'm wondering how we estimate them, like how do you get to know the emission error and the commission error? Like omission error, it looks like, more.
How you calculate this, you mean?
I mean like how you estimate them, like how do you figure that out? Like, because when you don't know, like
how do you do the predicted? How do we figure out the A, B, C, D?
No, I mean C and B.
C and B. Okay, so this is predicted absent when it's present and predicted present when it's in reality absent. So these are errors.
So it's like by resampling or sampling? Or
Yes, it's by sampling. So I have a sample of presence data, and then I build a model. Then I have a map of prediction, presence, absence, and I'll get to that next,
right.
How we get to the presence, absence. And then I map my presences and I map my absences, and those presences that are predicted absent will be the C, and the absences that are predicted present will be the B.
Right.
So we map known data, our validation or our testing data, we map that on the model output, model prediction of presence, absence. Yes, Christopher?
You do need that to be C divided by parentheses A plus C.
Yes, sorry, yes, yeah. The mathematician in the room, thank you. Well, one of the mathematicians in the room, but you're right, I should have had parentheses there. Thank you. Yeah, it's divided by the sum. Yes, Vitaly.
Area or a very specific area, can you do it per smaller subset?
You do for presence background, you do per smaller subset, the absence. And that's in the modeling algorithm that samples samples a fixed 10,000 background points, or you can change that. And that's in Maxent. We'll talk about that tomorrow. But yeah, it can be over the entire area if you want, or it can be over the area that was actually used to train, the samples that were used to train the model. Yes? Yes, so again, we need parentheses here, and yeah, that's a big problem. But what sensitivity and specificity show us is where the model is doing well. So this is a measure of good outcomes of the model. So sensitivity means the presences that were predicted present over known presences. So in other words, if I have 100 presences and 90 presences are predicted present, so I have A equals 90 and C equals 10, then my sensitivity is 90%. So it's a high sensitivity, it's a good model, it only wrongly predicts absent 10 10 presences. So this is, when we see a high number for sensitivity, it means that most of our presence data were predicted present because we are divided by the sum of predicted present versus predicted absent. Or in other words, we are dividing by the actual number of presences, right? So this is predicted present versus the number of actual presences. And if it's high, it means that most of my presences were predicted present, so that's good. Specificity is using the correctly predicted absent, this corner. It's using that number over all known, dividing that number by the all known absences. So that's why we have B plus D, so the division is the sum over the sum of B plus D, which is all my known absences, but some were predicted absent, some were present, some were predicted absent. See, it's already confusing. But this means if I have high sensitivity and high specificity, it means that the model is winning, the model is correctly predicting presence is present and absence is absent. If I have high omission error rate and high commission error rate, it means that the model is failing. It's predicting absent a lot of presences and present a lot of absences. Okay, anything else? Okay, so yes, what we want is low or zero omission rate, of course, because that's an error rate. Now with commission, as I said, we are somewhere in a bind there because we are using background pixels for absence data, but we know that some of those background pixels will be present, but we just don't have records of presence. Nobody has sampled those particular locations, or the species has not been detected. The species occupies those pixels. We are sampling them as background. We shouldn't be expecting them to be predicted absent, but because they are pooled in this background sample, they will be predicted absent. So commission error is an interesting beast to deal with when we don't have absence data, when we work with background. Because a commission error of 40% or 0.4, meaning 40% of my background is predicted present versus 70%, or 0.7, which is 70% of my background is predicted present, I don't know exactly where that limit is. So dealing with commission error rate is not that easiest when we don't have absence data. So I said presence absence. I said I said we have a map of presence absence, and then we figure out how many of my presences are predicted present, how many of my presences are predicted absent, in order to generate that omission error rate. But the models, the predictions are continuous probability of suitability, meaning values between 0 and 1, right? So I have a maxent output, a potential distribution map that is not binary, it's not presence absence. It just tells me this pixel has a maxent probability of suitability of 0.45, this 0.47, this 8.8. I don't know, how do I decide what's present and what's absent? So for that, we need to threshold the models. So we go from this probability of suitability, a map that has values between 0 and 1, to an actual presence absence by choosing a threshold. So the threshold, here I'm not showing how I made the threshold, but the threshold is selected, can be selected based on known presences. So here's my species ID, coordinates, and then for these presences, known presences, here are the model values, the suitability values. So maxent is telling me that this particular location has a probability of suitability of 0.005. This particular location for the same species, a probability of suitability of 0.1. So what I can do is basically say the simplest thing. I will do this. basically what we can say, the simplest thing is say, okay, I want all the training data, all the training presences to be predicted present. So in order for all the training presences, these locations to be predicted present, the threshold should be the lowest maxent value, maxent suitability value at which all my training presences, and I'm emphasizing training presences, not testing, all my training presences are predicted present. So in other words, what I'm saying is, okay, maxent, you gave me a continuous output. Now I want a binary output, presence, absence. I'm going to reclassify all pixels at this low value or higher as present, and all pixels below this value as absent, which in turn means that I go from this continuous output to this binary output, presence, absence, that will now have all my training presence data predicted present. So all my training data, presence data, will be associated with pixels that are dark in this, on this map. And it's training because that's the data. We are not, right now, we are not testing the model. We are using the training data to threshold the model, but then once we have thresholded the model, we can use our test data to map it and calculate omission error over the test data. Questions here? Yes. No, very good. And sorry for the mic.
Of the species in the area?
The probability, the continuous output,
Yeah, so
is not related to density. This is what a modeling algorithm gives you, and it is probability of presence or probability of environmental suitability. So the output will not be density unless you use something that is not presence background or presence absence. You use it depends on the modeling. Let's say this, it depends on the modeling algorithm. But in this context of presence absence, when we have locations of presence and background, so we do the environmental matching of presence and background, we will have a maxent probability of, let's call it suitability, but not density of data. But Town has his hand up, so please.
I'll just point out that there's a growing body of literature about a very interesting linkage between these models and abundance or population density. It's using kind of a trick, and it's not related to these specific probability or suitability values. Rather, it's related to distance to the centroid of the ecological niche, the fundamental niche. So there's all sorts of complexities involved. There was just a paper published that basically discredited the approach, and that paper in itself was quite flawed. So I think there's a growing body of empirical evidence that you can get a predictive estimator of density, but it's not straightforward, not enough time in this class to get to that literature, but we can certainly pass on the papers too if you're interested.
Okay, so I said the simplest way of thresholding the models is to say, okay, I want at minimum all the training data that were used to build this continuous probability of suitability or presence, all those training data to be predicted present, so I use the lowest presence threshold. But if you're interested in ways of thresholding your models, there's a whole another can of worms there, and you can threshold the models based not on just presence, but also based on absence or background. You can balance sensitivity and specificity, these metrics. So there are other options. Always think about what kind of data, what's your scenario, what's your BAM diagram, what's your scenario of data, because in my mind, using something like balancing sensitivity and specificity when I don't have absence data, so my specificity measure is how many of the background points were predicted absent. I don't know what's a good measure of specificity when I have just general sampling of the background. I don't have presence. So in my mind, I never use balanced sensitivity and specificity unless my graduate students have already used it because they are sick and tired of using just omission error, just lowest presence threshold. But I personally do not balance sensitivity and specificity unless I have information about the background or absence. So, but there are many options here, and some of these may be better suited for your question and data you have than just the simple approach. I want all my training data to be predicted present. Questions? Okay, so that's threshold dependent, meaning I have a continuous output, model output, a map of potential distribution that has lots of values, continuous output, and then I threshold that model. Now there is the threshold independent option, and those of you who have run models or have read papers, the star is the receiver operating characteristic, and the area under the curve of this receiver operating characteristic. And this is an analysis that I think dates back to 40s that was used by radar engineers, I think, I can't remember. Then it was imported in medical sciences for testing the sensitivity of diagnosis or tests, and then finally made it into ecological niche modeling, I think maybe early 2000s or late 1990s. Early 2000s probably. So here what we do is remember sensitivity and specificity. Draw your confusion matrix again. So here what we have is sensitivity over 1 minus specificity as the two axes. And then we have the diagonal, which is the basically random model. No matter what we do, we have equal sensitivity and we have equal chances of being wrong in terms of predicting presences or predicting absences. So it's like a random model. But then above this diagonal, we have increasing model performance. So the AUC varies between 0.5, below 0.5, that would be even worse than random. But 0.5 to 1, we never reach 1. We reach maybe 0.999, but not 1. So the higher the AUC value, the better the model is because it's predicting known presences with less commission error. So as we move on this axis, we are increasing commission error. So we go from, I'm predicting Southeastern Europe to I'm predicting the entire continent. So I have to get to predicting the entire continent to predict all known presences as present, my test presences as present. So the faster we get to predicting presences present with a smaller area, with a smaller commission error, the better the model is. So that's the star of niche modeling, ROC AUC. Now, we don't have specificity, again, in presence background, which is maxent. We don't have specificity measures because we do not have absences. So what we do is proportion of area predicted present or proportion of background pixels predicted present. So that's the one difference. We still call it 1 minus specificity, but in reality, it's proportion of background or area predicted present. So that's ROC AUC. Talking about limitations, took a big sigh. This AUC value depends on extent of my training region. So if I have the entire European continent and I'm working with a Mediterranean endemic, the AUC will be quite high because I'm training the model on an extreme heterogeneity of the environment when my known presences are limited to just the Mediterranean region. So extent of training region is a big problem in terms of inflating AUC values. So we get a high AUC and we assume, okay, the model is really good because I got a high AUC. And then species potential distribution across the study region also varies. Different models are never capable of having a 100% commission error. So some models are never able to predict the entire study region, where some models are. So that's another problem with comparing these AUC values across modeling algorithms. So one proposed modification that that happened when I was in grad school, so I worked with Town, was to look at only a portion of this curve, of this AUC curve, and then ignore performance at high omission, or do not count that portion of the curve that represents high omission error, because that is not what we are after, right? We want low omission error, so we shouldn't look at the portion of the curve that includes high omission error. And then make sure that both models you are comparing, or multiple models you are comparing, model outputs, general linear model, I don't know, a card, make sure that those models can actually predict the same extent. And so you have that proportion of area predicted present is constant, or not constant, the relative, the total area is constant, and then you have relative to that total area, constant total area between when you compare models. So this is what it looks like. Instead of taking the entire curve and calculating the AUC, the area under the curve, under this entire curve, we set a threshold of bad models, or a threshold of low sensitivity. And then we also truncate or discard the area of the curve where the model is not capable of predicting anything. So this means at 70% of the study area, that's the maximum distribution that this algorithm can produce. So I shouldn't count the entire area under the curve. I should count only that portion, because the model never gets to 100% commission error. And then with the sensitivity, basically we are saying we don't care about models that have 90% of the data predicted absent, 80, 70, 60. We don't care about those models that predict high omission error or low sensitivity. We are going for the highest sensitivity and lowest omission error. So that's the threshold over there. So then our area under the curve becomes just area under this portion of the curve, not the entire curve. Now this has not seen a lot of use. I mean, I've seen it on and off in modeling, in papers, and I've used it on and off, so it's not the thing to do necessarily. It's one alternative. But the most important part is to think about the limitations of these AUC scores, that we shouldn't compare them among species, because we have different when we have different training regions, especially when we have different training regions, it's really hard to compare AUCs, because we are using different extents and different heterogeneity of the environment to build the models and then test the models. But with the limitations, AUC is still used in all papers, or I don't know, most papers, and it is required. If you submit a paper, the reviewer will ask, so what was the AUC value of your model? So we are not abandoning, but we should be aware of the limitations. We should also include other measures of model fit or model performance. We should not rely just on one measure, ROC AUC. And another metric that has been used in recent years is this true skill statistic. And it's a sum of sensitivity and specificity -1. And here's where Xiao and I probably disagree. Well, for sure disagree. I don't use this because, again, what is my specificity when I don't have absence data? But I've submitted papers where, in one case, or a couple of situations where a reviewer said, you need to calculate TSS. I was like, OK, I'm going to calculate TSS, even though I will have to include a caveat. I do not have absence. Specificity is calculated over background samples. It's not true specificity, but there you go. I'm calculating specificity. The other thing is, this is also a threshold-dependent approach. So I already have this part as omission error. what I'm doing with TSS is I'm adding the commission error to my evaluation. So I already have one part of this metric as omission error. I don't have the other part of the metric. And it's a threshold-dependent approach. It's not a threshold-independent approach. So it has the same, it has similar, or it has limitations just like omission error, which is why we cannot just report one metric. We should report multiple metrics, but yes. So TSS is now becoming quite widely, it pops in literature almost, I think almost all papers that I've read recently have TSS in it. And I think that's all. Any questions? Yes. Oh, sorry, microphone, whoever had the last time.
So say if you want to model any species that belong to South America and you blindly use South America as your calibration thing, then your model will always have a great AUC, basically, because the extent is so big.
Exactly, yes.
That
so
your model will be, you will say it's great, but maybe it's crap.
It's
potentially.
It's less useful if it predicts 90% of South America, and you have a species that is, restricted to the Amazon basin. So, yes, my answer would be: do not use the entire South American extent if you know that your species is an Amazon basin species. So train the model to the best of your ability or knowledge, the known range of the species. If you don't, and for a lot of species we have no idea what is a reasonable range. We can go to IUCN and download range maps. If we have absolutely no idea, what we can do is use the data we have, the presence data we have in hand, to generate a minimum convex polygon and then buffer that by, let's say, 100 or 200 kilometers, and that becomes our training region, meaning that we are feeding presence data at the extent, with environmental data at the extent of known presences. And I say buffer, and we'll talk about that in the afternoon. I say buffer because if you use just the minimum convex polygon of your known presences, you will not sample the environment beyond where the species was reported present.
And should that match the accessible area for the species?
Definitely. If you have, so that's where M comes into play. So when you think about, okay, I'm going to run models, how am I going to do this? Think about the BAM diagram. What is the M for the species? And Town was saying, these are not easy questions to answer, and they are interrelated. So M may depend on B, right? If it's a species dispersed by a frugivorous bird, right? I need that. In order to figure out M, I need to figure out the B, the biotic interaction that makes dispersal possible, or M. So yeah, these are serious questions. Did you have a question?
Yeah, are the models assuming contiguous regions?
You mean if I have South American continent, it's one contiguous region? Yeah. Yes, yes. If I define, the study extent is South America, then it will sample across South America. If you want disjunct distributions, then you have to mask out, let's say it's, this is not biologically correct, but if you have a species that is in the Amazon basin and Atlantic forest, what you want is those two disjunct distributions to become your one training region, not entire. If you give it the entire South American continent, it will sample the entire continent. I think was that the question, or?
More or less, you answered it kind of secondarily, but if you have a training data set, you can apply it to a second region, basically.
Oh yes. So that's model projection, and we'll talk in in all modeling approaches. That is one important application of models, right? We have a training region, we calibrate a model, and then we take that model and project it the entire continent. We projected, maybe we trained the model in Europe. It's an invasive species. We projected on North America. Yeah, so that's.
Or even that could work in little micro regions too, right?
Okay. Yes.
Thanks.
Yeah. So maybe you have you have a hunch that the species should be present in these other areas, but you have no data. You can train the model at the extent of known presence, and then apply the model, project the model to those disjunct areas, population, possible population, meta-population, I guess, or subpopulations in a meta-population approach. Subpopulations, yes, in a meta-population approach. Okay, so you're gonna defend the TSS. You get a whole afternoon tomorrow to dismantle everything that I just said.
For the TSS, I think
Told you.
I agree with you. The specificity is not true specificity because we're using pseudo absence instead of true absence. However, I think for AUC, for the other evaluation metrics, they all rely on this fake specificity.
You're right.
That's my
It's
comments on that.
very good point. TSS is no better or no worse than AUC, because AUC is also using specificity right here.
So
And-
it's okay to drive 90 miles an hour if everybody's driving 90 miles an hour.
No, not at all. I'm all about the rules. Yes. do not drive 90 miles an hour, and do not rely on just AUC or just TSS or both of them. I do include, I include omission error. I know it's more limited in terms of what we are measuring, but I think omission error is still a good metric.